you should see.
Yes,
should see my screen now, and
we see it. Great.
Hopefully only my the portion of my screen where my presentation is. I have a large screen, so don't want to share more of that than necessary. I would like to thank the organizers for a kind invitation. It's the first time I do something like this, and but it was clear to me that that this was a good way to contact other people with similar interests and share some of the findings that we've had the last couple of years. So without any more ado, I am Øyvind Halskau from the University of Bergen. I am professor at the Department of Biological Sciences. I will give a seminar now entitled Nanodiscs for In-Solution Kinetic and Steady-State Investigation of lipid effects on alpha-synuclein, misfolding, and aggregation. And I will try to get a laser pointer here, a virtual laser pointer. Is this good?
Yeah, looks good.
Very good. So my interest is in protein membrane and protein lipid interactions and the mutual effect that proteins has on lipids and the effect that lipids have on protein. And of course, the biological consequences of this. So the artwork here in the background, prepared by Carl Bryn, doesn't have any direct to do with what we're going to talk about today, but it is related to protein membrane interactions. I wanted to show it because Carl Bryn makes such great artwork. So, misfolding in neurodegenerative diseases. So when a protein folds, as many of you know, the protein has to deal with a lot of challenges, such as translational errors, that there is a crowding in the cell cytosol, up to 300 to 400 milligram per milliliter protein concentration. That's quite a lot. There are large proteins for prokaryotes. You have an average size of 34 kilodalton, bigger for humans or similar organisms. Many of these are intrinsically disordered proteins, or they have a weak fold. They don't fold strongly, meaning that they're prone to perturbations and maybe also then misfolding. Protein misfolding, according to Palchin et al., affects 1 in 7 proteins in the cell. That's quite a lot. So, of course, there is a machinery in the cell to try to nudge things back into onto track. So here is a properly folded protein. If it misfolds or aggregates into metastable state, there might be chaperones nudging it back in this direction. If this persists, you can get amorphous or oligomeric or fibrillar aggregates. And of course, there might be mechanisms for taking this back on track again. But as this proceeds, it gets harder and harder. Eventually, of course, you can have clearance by degradation, but some aggregates, especially this, is quite resistant to this. So there's clearly interest in investigating these matters and what nudges things in the right direction, what nudges it in the wrong direction. So folding of proteins is essentially a search for an energy minimum in a landscape, and an energy landscape where points on this landscape reflects folding or free energy and some conformational fold. So the native protein would be here. Folding states that are on track would be here, but it could also go onto, well, let's call it the dark side, since there is a dark shading here, and form oligomers, amyloid fibrils, or other aggregates. So what if control of this fails? Well, you can get a number of diseases, as I'm sure you all know. Some of these are neurodegenerative in nature. Here is a selection of which parts of the central nervous system is affected in different diseases. And these diseases are generally characterized by having loss of neurons, neural connections, and gain of motoric symptoms. There are risk factors. age, lifestyle, persistent states of informations, and of course genetic variations or downright on-off inherited states, such as especially the Huntington's disease. So the rest of the talk will focus primarily on Parkinson's disease, or rather alpha-synuclein, the misfolder in Parkinson's disease. So misfolding and so-called seeding is a shared feature of many of these neurodegenerative diseases. So in the molecular theory of this, one assumes that misfolding can happen spontaneously, but it can also be seeded. There might be some particle or some molecular state nearby that promotes the misfolding and promotes the sticking together and aggregation of multiple units of misfolded protein. And if this process gets going, for instance, by the misfolders promoting more misfolding, then you could get a runaway state. Here is a schematic representation of this for Alzheimer's and Parkinson's. Okay, so that was a little bit of background. I hope it's worthwhile spending some time on it. So several years ago now, actually quite some years ago, I was interested in the following. What if the lipid membrane could act as a seed or a promoter or a catalyst even of misfolding events? Surfaces independent of the nature are often act as catalysts. So what if the membrane could be such a surface? Admittedly, a fluid and very dynamic surface, but still an interface at least. Lipids might actually help proteins fold, so why can't they also cause misfolding? Many proteins unfold slightly when contacting the membrane or fatty acids or other lipids present. And what if this slight unfolding kind of primes them for misfolding, or if there is another protein next to it on the surface that kind of starts the ball rolling. And for the connection to potential risk factors, lipids are linked to aging and lifestyle, and this could, if you speculate about it, provide a possible unknown molecular mechanism between misfolding diseases and risk factors. Okay, so after many false attempts, I got funding to pursue this from the Norwegian Research Council. The project started in 2015 and ran to last year. And one of the papers that came out is what I will present now. And that was also the basis of the invitation to this event. So the title is Cholesterol-Containing Lipid Nanodiscs Promote an Alpha-Synuclein Binding Mode that Accelerates Oligomerization. Now, what follows is just 5 small graphical icons where I give a bit of an overview of what we will discuss next. So first, I will spend some time on our choice of lipid model system. Then I will present what kind of lipid nanodisc systems we produced for this study. Then I will show you some results which I think are nice, where we're able to associate misfolding and aggregation behavior to cholesterol that accelerates this process, and to fluid membranes which actually suppresses it. I will also show some AFM slides where we can see the nanodiscs and maybe also some aggregation events taking place near them. Finally, and this was a pet peeve for me when doing this work, I wanted to link aggregation and misfolding events to changes taking place in the protein. I wanted to see what happened to the protein fold when it was in a state that was prone to misfolding and aggregation, and compare that to states that were less prone to do so. Here we also use nanodiscs and in combination with NMR in an in-solution steady state experimental setup. Okay, so why SMALPS, the choice of lipid model systems? Well, as an NMR spectroscopist and a spectroscopist, I like nice solutions that don't aggregate, that last for a while in solution, that have no very big aggregates, because this interferes with many spectroscopic techniques, including NMR. So liposomes were not the system of choice right from the start, because I wanted, although it's possible to pursue this with NMR, you're forced to do very indirect studies then. So nanodiscs are much smaller than liposomes and therefore much more attractive in these days. They provide monodisperse and relatively stable solutions, and there is no significant exchange between the disc components and the solvent. That's also good. And of course, we were interested in the planarity of the disc bilayer, because a lot of things have been done on alpha-synuclein aggregation in the presence of curved membranes, but there is less, at least when we published this, it was less papers that studied this using planar bilayers. So nanodiscs could be scaffolded by proteins, as I'm sure you know, but the reason why we ended up with SMALP nanodisc was that in our hands and view at least, it offered even better solution properties. And alpha-synuclein would then be the only protein component present in the sample, which was good. And the membrane scaffold proteins here, they are actually distantly, vaguely related to alpha-synuclein. So it was really good to have that out of the way. So therefore, we went on with SMALPS. So we wanted fluid bilayers with and without cholesterol. So we made vesicles, added SMALPS in the concentration which are logged here. And then the major nanodisc size fraction are given as follows. We also assessed the average mass fraction of lipids that were converted from vesicles to nanodiscs. So as we generally we experienced that making nanodiscs from DOPC vesicles were really easy. They readily went into the nanodiscs. Highly fluidic spherodionic lipids, they went right into it with high conversion efficiencies. The same with cholesterol, yielded much lower mass conversion. And we saw the same with our lipids, which convey a net negative charge to the bilayer. But these again were maybe intermediate between these two. So we had some optimization before we arrived here with respect to inclusion of cholesterol. At the time, at least, we couldn't find anyone else who had tried or reported inclusion of cholesterol in nanodiscs. So we were kind of hoping to be able to report that as a small first, but maybe we were wrong. I don't know. Maybe you know. So here is our final 4 model systems. One of the things that we observed, cholesterol induced this size shift as shown here. This is dynamic light scattering results. And the final SMA nanodisc properties were assessed by size, setup potential, and compared to the vesicle setup potential. So these are the parent vesicles. We can see that there are some changes here. So now that we had the nanodiscs, we were ready to do our first misfolding aggregation assay, where which we essentially performed in the well plates, and we used a well-established fluorescence assay called THT aggregation assay. So I think that many of the participants here may be familiar with it, but still. So when you have aggregation into fibrils, the fibrils have a high affinity for THD fluorescence probe. So then you easily report on the amount of fibrils in the sample. However, the onset of these processes is very stochastic. So we essentially did one small experiment in each well plate, and then we assess that stochastically. So what we have plotted here is wells that has started to aggregate. So we don't plot individual wells, we just ask for each well, has it started? Does it report THD above a cutoff, or doesn't it? And then we got, we believe, very nice curves that allowed us to circumvent some of the difficult and random aspects of these kind of assays. So the blue line here is alpha-synuclein only. These two have no cholesterol present. They are fluid, highly fluid bilayers, and these two have cholesterol present. So we just by eyeballing this, we see that the cholesterol produces a strong shift sorry, this should be, of course, be the other way. I'm sorry, the barrel should be pointed the other way. My bad. In any case, these traces have no cholesterol. These have cholesterol, so we have a strong early onset of aggregation in the presence of cholesterol. We could analyze, or we could fit this data to what is called the Finke-Watzke two-step model, and there are three important parameters here: the half-time of fibrillation, which is here, the lag time, which is related to when this curve starts, and the growth rate. So if we do this analysis, we get the following results here. And here we see that the cholesterol really reduces the lag time, and it really increases the growth rate. So we were very happy to see that result. In parallel, we took AFM images of the samples associated with the lag phase of the fibrillation. So just before we believed the aggregation would kind of take off. So here are some representative slides. This is alpha-synuclein only. We see that there is a lot of well, small granular deposits. So alpha-synuclein clearly aggregates. If we zoom a little bit in, we see the start of fibrils. Remember, this is before the lag phase. Now the same situation, but in the presence of nanodiscs, we see the nanodiscs. These are the clear white spherical entities here. We see less of this background, but of course there is some, but much less. And we see some kind of aggregation that seems to be associated with the discs. Then if we add cholesterol and look at this again, we see a completely different picture, a lot more aggregation overall. And in the insert here, we see a quite long fibril. So that was generally in line with what we had observed in the previous aggregation experiments. And then for the other cholesterol state, but with the net negative charge in the membrane, we also see a lot of different morphologies, but maybe shorter fibrils or a slightly different background. But very often we see that the protein aggregation sort of seems to grow out of the nanodiscs, which confirmed that this was a effect that took place in presence of the lipid model system. All right, so then we went on to try to explore what fold of or what folding state of alpha-synuclein might be associated with accelerated aggregation in the presence of lipids. So alpha-synuclein is an intrinsically disordered protein. So if you run an NMR on it, you will not see native fold. You'll see a pretty collapsed fingerprint. For those of you who do NMR, you will see this immediately. And then we took these fingerprints where each cross peak is essentially one correlation between a backbone proton and a backbone amide. And we did the same experiment at constant alpha-synuclein and different protein nanodisc ratios. And then we looked for shifts in the peaks or changes in peak volume. So this is a in solution reversible interaction experiment. And the timescales are such that we, rather than seeing shifts, we observe a loss of peaks or a loss of peak volumes, if you want. So we plotted the peak volumes and made graphs like this, where we the fitting gave us an dissociation constant for each residue that we could identify. Clearly, we couldn't identify all residues in alpha-synuclein, but enough to sample the entire length of the protein. This was, of course, not a full dissociation constant, but an apparent dissociation constant that we could relate to different parts of the protein. So here are just 3 examples of different curve fittings picked for being high affinity, intermediate affinity, and low affinity states. And then we could plot the results for different lipid systems like this. And for each residue here, we could give a color code for apparent dissociation constant. Then we could relate this to different regions. Here is the N-terminal region, which is associated with membrane binding. The NAC region is associated with, or it's called a non-amyloidic component region. But it's actually this one which appears to be active in misfolding events, and the C-terminal region, which is has more negative charge and is thought to kind of provide a little bit of solubility and maybe contribute to keeping alpha-synuclein out of trouble. So we saw that there were differences between these 3 regions, which are defined by literature and previous work of many people. So that was already interesting. So and then we compared cholesterol and no cholesterol. So we looked for changes induced by cholesterol. So if you look at these bar plots here, we see that the change in the apparent affinity constant when cholesterol is present for DOPC is quite large. For the other system, DOPC, PE, PG, we have less change, but clearly, or at least in this this green region here. So based on this, our argument was that in this case, in the case of the green regions, we see either large lowering in affinities or no lowering, while the other regions are actually changed. So our interpretation was that even though cholesterol reduced the overall affinity of alpha-synuclein, it did still associate when cholesterol was present. And here comes the important part: when cholesterol is present, that forces alpha-synuclein to rely more on the NAC region for binding. So there's a shift in binding mode. And our interpretation was that as alpha-synuclein attached to cholesterol bilayers, the association became more superficial. The protein was not able to penetrate that much, maybe, and it had to bind with this region. As more alpha-synuclein bound close to this, the polypeptide choose to interact with each other instead of fully embedding or properly interacting with the membrane. And that was our interpretation of this results that would lead to the observed acceleration in alpha-synuclein aggregation. So our main conclusions, just to sum up: cholesterol is more difficult to include in SMALPS, and you get size effects, so you get larger nanodiscs. Nanodiscs proved to be excellent tools for in-solution studies of reversible protein membrane interactions. Fluid membranes suppress misfolding and aggregation, and cholesterol speed up aggregation. And we were, by combining nanodiscs and NMR, able to suggest cholesterol-induced binding mode that would explain the accelerated aggregation. Now for my final slide, I just have acknowledgements. These are actually, so Martin Jakubek did fantastic work on this paper. He was my PhD student until fairly recently. He did, all the experiments, performed it really well, designed and redesigned the approaches as was necessary, and essentially just did a fantastic job on it. Aurelia is a close collaborator. Vinit George contributed with samples. Samuel Furs was a postdoc in my lab at the time. He introduced us actually to the SMALPS nanodiscs. Diana did SPR and sample preparation. Espen did experiments and helped Martin every step in the way, and also, of course, interpreted data. Jarl Underhaug, valued collaborator in NMR, and Ludmila Mirosova-Roche, also a very valued collaborator. This is not a complete set of pictures. I'm sorry, couldn't dig up everyone, but I would also like to acknowledge Igor Yashisin for the AFM work. And Morten, he did, not depicted here, Morten did the alpha-synuclein expression, designed the expression system. I would like to acknowledge University of Bergen and my department for supporting me, and the Norwegian Research Council grant that allowed us to do this work, and the Norwegian NMR platform in Oslo and Bergen, and the associated infrastructure grant. Yeah, and it's here's my wife. She was not on this paper, but she has a birthday today, so congratulations, Milo. I hope you see the YouTube video. That's all I have to say. Thank you for your attention. I will take questions.
Thank you so much. So is there any questions for? Yeah, Anna, go ahead.
Yeah, I've got a couple of questions. Fantastic talk. I really enjoyed the data, particularly your very nice AFM data. I was just wondering if you had considered or had tried negative stain EM or even query EM for looking at the aggregation, because particularly, yeah, for negative stain, it might provide quite an easy quantification of the extent of aggregation that you saw in different conditions.
No, we haven't tried that, but that would be a good, let's say, control or an additional quality check, if I understood you correctly. Could you give those reagent names again?
Yeah, just a negative stain EM. I think it would be quite
negative stain EM. Yes. No, we have not do that, but that could be another way to get images of course.
Quite an easy experiment, probably easier than AFM.
Yeah,
also,
well, we were offered, Igor offered to do AFM, so we were very happy to pick him up on that. But EM, yes, of course, it's a good good way.
I'm sure that there's lots of people in Leeds who would be happy to collaborate if you wanted people to image that for you.
Great.
I also had a question about, I don't know if this is even possible, but did you try cholesterol alone nanodiscs?
Oh, so a 100% cholesterol
Yes.
thing. Well, the way we made the nanodiscs, we had first to create vesicles, and I don't think I could make vesicles with 100% cholesterol. I don't know. I assume that would not work, but I've never tried it. I've never seen anyone do it. But that. But there might be possible to maybe if nanodiscs could kind of extract or include cholesterol from something else than nanodiscs, very concentrated solution or suspension of cholesterol, maybe that would be something. That's interesting. I wouldn't know how to do it, but it would of course be a challenge, an interesting challenge, and a useful outcome if we could have something with pure cholesterol. I can, if I may, comment a little bit about cholesterol. So we prepared the vesicles, the parent vesicles, with 30% cholesterol, and we verified that cholesterol had indeed entered the nanodiscs, but we did not quantify it well. That was difficult to quantify it well. So it may be that our nanodiscs have less than 30% cholesterol, but we do know that it has a decent amount at least. So in our hands, cholesterol sort of made it more difficult to make nanodiscs.
Yes, absolutely. I think possibly my question about cholesterol only is probably easier if you're doing the MSP nanodiscs, where you can just kind of add in your lipid of choice rather than the SMALP nanodiscs where you're working from vesicles first. I wonder then, going from your point about not knowing the percentage of cholesterol, whether you could be having different amounts of cholesterol in your, was it DOPC alone or PCP-GPE with cholesterol, and whether that might account for the fact that you don't see the same patterns in the NMR. Do you think that's possible, or do you think that probably roughly the same amount of cholesterol is incorporated?
I think we don't, so to be brutally honest here, we don't have full control of the amount of cholesterol, cholesterol incorporated. So what you say is completely true. DOPC and the more complex mix might include cholesterol differently as they are formed. And we see that they're not the same. They don't have the same size on DLS. They don't have the same setup potential. We did think of preparing another study where we took DOP3 100% and then added more and more cholesterol until it stopped, and then doing a better job at quantifying cholesterol in the final nanodisc, and then see if we could get some dose dependency out of it. That would have been a lot of that would have been an entire paper and a lot of work. So, and you know, time and money just runs up. Does that answer your questions, or did I steer off into a different path?
No, that answers the questions. Thank you very much.
You're welcome.
Yeah, go ahead, please.
Yeah, thank you. That is really interesting. I really enjoyed your talk.
Good to hear.
I was wondering, in one of the first slides, you showed a table with the conversion of lipids. So the percentage of lipid that is transferred from vesicles into nanodiscs. How did you determine that?
Yes, I will try to find the correct slide. Is this it?
Yeah, that's the one. So in the last column.
Yeah, so you're essentially asking about how we got to these numbers?
Yes.
Yes.
Yeah.
So again, I have to credit Martin Jakubek here, who devised a way to do this. What I believe, so we used mass spectrometry, so what we did was for this was that we collected we ran the samples on gel filtration, and we collected the vesicle fraction and the nanodisc fraction.
Okay.
And then we did mass spec on the lipids, and then I think he essentially counted the hits. And benchmarked it so that he got a picture at least about how much mass was transferred from the vesicle fraction to the nanodisc fraction. I believe this was how he did it. Did that answer your question?
Yeah, it did. Thank you.
I can get more details if you need.
No, I think I got the concept. Yeah, that's good enough for the moment. But thank you. And happy birthday to your wife.
Yeah, thank you. Was that too cheesy of me?
No,
I was very busy this week, I just wanted to show that I could spend some time on her at least.
Great. Okay, thank you.
Any other questions for? No. I have a naive question, if I may.
Of course.
How difficult is to address these lipid-protein interactions, particularly for alpha-synuclein and lipids or cholesterol in vivo, for example, in the cells, or?
How difficult it is to I'm not.
For example, if for example the interaction between alpha-synuclein and the lipids that can
how they can be followed up in vivo.
Yeah.
Yeah. Well, for cell systems at least, you could stain with antibodies with the GFP tag and locate it that way, and at the same time you could use a cholesterol sensitive stain. And then if you're clever in cell biology and microscopy, which I cannot say that I am, but I think it would be possible then to assess colocalization in this way, especially if you have a confocal at hand. I think there is a cholesterol specific stain. I can't remember its name, but I think this could be done. That would be for cell biology, and of course you could, I guess, section patients as well if you had patient biopsies present. Does this answer your question?
Yeah, thank you very much.
That of course would be very interesting to link this data to cell biology and organisms, and that would increase the relevance of what I'm doing. So I'm of course interested in going that direction, and I think I have to go that direction to get funded too. It's yeah.
My other question is how long you incubated the disc with protein. So is it like 5 minutes overnight, like a specific condition?
To get aggregation?
Yeah.
These experiments are quite long, so you have to be patient. So for alpha-synuclein alone, at 24 hours, you're halfway through the aggregation experiment. So yeah, so we had to be a bit patient here. And there were many trials and error before we came to that graph. But yeah.
37 degrees, right? That's like body temperature or room temperature, or
the temperature at which we ran these assays?
Yeah.
I think it was room temperature or 25 degrees. That I would have to check, but it's all in the materials and methods, I suppose. I think there is another hand, virtual hands.
Yeah, that was Anna already asked it, unless.
Oh, yes.
Other questions. Thank you very much. Other questions? I appreciate that. Excellent talk.
Thank you so much. Thank you all for asking interesting questions and inviting me and following the talk.